Good morning or good afternoon. My name is Mary McCabe, and today I'll be giving a short presentation regarding some of my work titled Exploring the Development of Innate Immune Responses to Infection in Early Life. And so just to provide a brief overview of what I'm going to cover today in my presentation, firstly, I'll introduce early life viral infections and explain why they're a major global health problem, particularly for young infants. I will then describe my hypothesis, which forms the basis of this submitted abstract and presentation. I will then provide some information regarding the methodology used and my results, and finally provide some food for thought and future perspectives that need to be addressed to move forward within the field. But firstly, let me introduce myself. My name is Mary McCabe, and I'm a first-year PhD medicine student at the Wellcome Wolfson Institute for Experimental Medicine at Queen's University, Belfast, Northern Ireland. Primarily, I'm a wannabe molecular virologist and immunologist, interested in pediatric viral infections and innate immune interactions in early life. However, through my experience as a hands-on wet lab virologist, I have come to appreciate the necessity of translational bioinformatics within my work, of which will be a major discussion point of my presentation today. You cannot really publish data without bioinformatics, which forms the component of any well-written paper or well-designed experiment in the modern era of virology. And so today I'm going to present a bioinformatic component of my research work. And so, without further ado, let's discuss early life viral infections. Respiratory tract infections represent a major cause of childhood morbidity and mortality in both industrialized and developing nations. Most children, when they get infected by a virus, experience an uncomplicated upper respiratory tract infection, most commonly infection of the nasal epithelium, which lines the nasopharynx, which is normally self-limiting and doesn't really require any treatment. However, a minority of children will develop severe acute lower respiratory tract infections. And despite systematic epidemiological data identifying many associated risk factors, the mechanism by which a minority of children experience or develop severe acute respiratory tract infections remains unknown. Furthermore, despite the airway epithelium being the main target of and the front line of defense against viral infection, there are limited investigations which examine tissue-specific responses within these tissues. And so, why is this important? Well, chronological age is a major risk factor for respiratory viral infection. Children less than 2 years old experience more respiratory viral infections, which are often more severe and result in more hospitalizations compared to older children. Furthermore, as the peak of viral disease occurs early in life, it's likely that non-specific innate immune responses to infection have a greater significance during early life viral infection. And therefore, it is hypothesized that it is likely differential early life innate immune responses to viral infection may determine infection outcomes in those children who develop severe disease. And so to put a bioinformatic twist on this hypothesis, we hypothesize that innate immune responses to infection within respiratory tissue develop with chronological age, independent or irrespective of the infecting virus. And to address this hypothesis, we assess the possibility of using in silico approach based on the identification and reanalysis of published datasets. So, as a classically wet lab trained virologist, there are many different approaches of what I could have taken to explore such a hypothesis. And it seems obvious that I could collect samples from infants infected with different respiratory viruses and assess gene expression. However, to fully appreciate and assess the wealth of data that is contained within our public data repositories, I realized that the best approach for this analysis would be an applied translational bioinformatic one. And so I jumped into headfirst the world of translational bioinformatics to utilize an in silico approach to provide information regarding a major in vivo problem. And so we designed a simple workflow incorporating publicly available, accessible bioinformatic tools to explore our hypothesis. So first, a search query was designed for each identified virus. Inclusion and exclusion criteria were then outlined to aid in the identification of appropriate datasets. The search for each virus was then conducted in two of the largest public repositories for functional genomics high throughput data, Gene Expression Omnibus, or GEO, and Array Express. Identified datasets were then screened for appropriateness, and further analysis was then performed using the NCBI tool GO2R for differential expression. 7 common pediatric viruses and coronaviruses were created within our datasets. These viruses were chosen for their commonality in infection during early life and their ability and frequency for being associated with viral-related disease in young children. Of course, coronaviruses were also included in this query as per their current relevance and explosion in gene expression data available. A search query was then systematically designed incorporating 3 concepts: respiratory epithelial cells, early life, and the infecting virus. And all of these search queries were designed utilizing control vocabularies. The inclusion and exclusion criteria were then designed to filter out the most relevant and appropriate data for further downstream analysis. So some inclusion criteria include: studies were restricted to those conducted within primary human airway epithelial respiratory tissue and completed on total RNA-seq or microarray platforms. Only children less than 2 years of age, the age group who experienced the greatest frequency of viral infection, were included in this analysis. Some inclusion criteria included those studies which explored responses between acute and convalescent or mild and severe infected children, as mild and convalescent states of infection are not representative of non-infected states. Data analysis was then performed using the publicly available interactive web tool GO2R. This online tool allows the user to compare experimental groups of samples from a GEO series to determine differential expression between different experimental conditions. As a wannabe virologist/immunologist, I am learning to be a power user of the tools developed by bioinformaticians for the wider bioinformatician community. Then, once we decide on our groups that we're going to compare with their differential gene expression analysis, GO2R fits a linear model and conducts comparisons on supplier-submitted data between samples using the bioconductor package Lima. And so, the results. Was there any data available in order to conduct my analysis? And so we searched using our queries within public gene expression repositories, applying our inclusion and exclusion criteria in a systematic fashion to identify datasets which we could use. And I expected for such a major prevalent cause of childhood morbidity and mortality, a cornucopia of data would be available to complete our analysis. However, as was the surprise to us after completing a systematic review of the data that was available, only one dataset was identified that could be utilized to attempt to answer our hypothesis. This study compared nasal and blood samples to evaluate host transcriptic responses to infection in young children, generating gene expression data using Affymetrix human clarion D microarray strips. And so just to provide a brief insight into the design of this original experiment, this investigation collected cells from the nasopharynx using a mid-turbinate flocked swab. Filtering out the data that was not relevant to our search query and those that did not reach our exclusion and exclusion criteria, identified 3 groups of samples appropriate for further analysis. 5 symptomatic cases who were infected with rhinovirus, 6 symptomatic cases that were infected with respiratory syncytial virus, and 3 asymptomatic control patients. Further information regarding how the samples were processed was located within the publication supplementary data. This revealed that the data deposited within GEO as the finalized and combined series matrix file had undergone RMA background correction, medium polished probe signal summarization to probe set levels, and quantile normalization. This information is useful in helping to identify what further processing must be completed within GO2R to complete my analysis. And so, moving forward, 3 groups of samples were identified as relevant for our analysis. A control group, which contained 3 uninfected samples, a rhinovirus infected group, which contained 5 rhinovirus infected samples, and a respiratory syncytial virus infected group, which contained 6 respiratory syncytial virus infected samples. However, before moving forward, there were a couple of important characteristics within our data that first must be mentioned. Firstly, the control or mock population only contains a minimum of 3 samples and possesses an overrepresentation of males compared to females. A further comment relates to our infected populations, in which infants infected with respiratory syncytial virus are on average much younger than those infected with rhinovirus or our mock or control populations. These facts become relevant when we attempt to assess differential expression with chronological age later on in the analysis. So GO2R allows the user to define groups and set the parameters of each analysis. So for my analysis, Benjamini and Hochberg was used to correct for multiple testing, as it possesses an intermediate level of statistical stringency, with statistical significance being set for an adjusted p-value of 0.05. Further options, such as log transformation, application of Voom precision waves, or forced normalization, was not applied in this instance, as the deposited series matrix file was already processed and normalized and then deposited in GO2R. And so, using the 3 previously identified sample groups and our GO2R parameters, I ran the analysis within GO2R. GO2R produced a box plot in which the medium of each of the sample probe intensities was relatively consistent across all samples analyzed, with no obvious outliers present, suggesting the data did not require further normalization. To check the separation of our samples by conditions, GO2R also produced a UMAP plot. From this plot, you can see, in general, control and infected samples did not cluster together. There was good separation between control and RSV infected samples. However, rhinovirus infected samples were more variable in their separation from control samples. And so first, I wanted to explore if I could use this dataset to conduct differential gene expression analysis between mock and infected samples, which would provide an indication of the suitability of my data for my analysis. So when we compared RSV infected samples with mock samples or control samples, GO2R called 1,337 statistically significant differentially expressed genes. Displaying this data as a volcano plot, the upregulation of classically associated interferon-stimulated genes, such as IFIT1 and IFIT2, was present in infected samples, and the upregulation of airway epithelial-associated transcripts within control samples. Similar to differential expression with respiratory syncytial virus infection, when we compared rhinovirus-infected samples with control samples, an upregulation of ubiquitous antiviral-related genes, such as ISG15, was present, and a downregulation of transcripts associated with epithelial cilial function was also present in infected samples, more than control samples. However, rhinovirus infection versus control samples resulted in much less statistically significant differentially expressed genes. Therefore, it is established that from this geoseries, it is in fact possible to assess differential expression of primary airway epithelial-derived material using GO2R. However, this was not the main objective of this study. Instead, we want to assess the differential expression within primary airway epithelial-derived tissue with chronological age. And so, can this differential gene expression be achieved with data that is currently available? And as previously hinted, this is not possible. The design of our original search query was to identify all those series and samples which may have aided in our identification of appropriate datasets for the exploration of our hypothesis. However, what is clear from these samples is that without clearly planned and specific experimental designs prior to sample acquisition, this hypothesis cannot be addressed using this data. Ideally, at least 3 control and infected samples would be available from infants in the same age group, or even in a more perfect world, paired and control mass samples for each child obtained at different stages during the first years of life would be available in order to conduct differential gene expression analysis. However, unfortunately, within this dataset, because of the varied distribution of ages within our mock or control samples and our rhinovirus infected and respiratory syncytial virus infected samples, it is not possible to segregate samples into meaningful groups to allow for comparison with chronological age. And therefore, analysis to explore the development of innate immune responses with chronological age is not yet possible with the data that is currently available. However, it's important to note that GO2R does possess some limitations and caveats. And in fact, when we further look at the data produced via GO2R, there are many instances in which the tool could have been improved for future use. So for example, when examining the genes that are differentially expressed between control and infected samples, Surprisingly, we found a positive control probe intensity value appearing in the list of statistically significant differentially expressed genes with infection. Of course, the inclusion of control probe intensities within the final data for differential expression most likely results in high level of background noise generation, which would make it difficult to identify true differential expression. And therefore, despite GOTR being an accessible, translational bioinformatic tool for non-specialists, it possesses critical caveats which must be considered before utilization, as demonstrated in this analysis, as these caveats and limitations can drastically impact the interpretation of generated data. So, for example, GO2R cannot perform any further processing other than what has been submitted by the supplier, including removal of outliers, removal of control probes, or filtering no or low expressed probes across arrays. GO2R also does not possess the function to compare differential expression between different series deposited within GEO and cannot pair samples within an analysis. So despite the accessibility of design, specialist knowledge is still required to aid in the interpretation of data uploaded and analyzed using GO2R. Thus, GO2R is an accessible tool for non-experts. However, its oversimplification of the differential gene expression pipeline may introduce confusing and misleading results. However, moving forward within this field, the rate-limiting step preventing meaningful data interpretation is the availability and accessibility of data. As mentioned previously, as a traditional wet lab virologist, there were different wet lab approaches I could have taken to explore this hypothesis. However, modern virology goes hand in hand with bioinformatic technique. So, for example, within our group, we have an excellent experience of working with viral infection of primary airway epithelial cell cultures. So here are just two papers that we published within the last year, which utilize these complex and specialized in vivo systems. However, the core findings of each paper was achieved using bioinformatics, emphasizing the critical need for translational bioinformatics within my field of research. And so to bridge the gap between the two disciplines, I took a translational bioinformatic approach to this hypothesis, defining a clear clinical question in which my findings of the analysis would have clear clinical impact. And that is what is most important. In order to produce results that have a clear clinical impact, one must form a clear clinical question, maintain a balance between bioinformatic tool accessibility and data availability in order to effectively inform clinical practice. However, as within our case in this bioinformatic analysis, despite processing and possessing a clear clinical question, an imbalance between the tools that are accessible and limited data ensures that it is much more difficult to make observations and conclusions that inform clinical practice and ultimately improve the care and the treatment of affected patients. And so, to summarize, despite respiratory viral infection constituting a major cause of childhood morbidity and mortality, there is a lack of respiratory expression data to explore innate immune responses to infection in early life. Future data generation and availability are promptly needed to address this issue. This presentation is a formal call for data generation to address the unknown mechanism of viral hypersusceptibility in early life. Finally, we may have the tools, but these are always meaningless without the data. And I would just like to make some final few acknowledgements. First, I would like to thank my supervisors for all their help and guidance and encouragement throughout my program. I would also like to thank the funders of my project for being funded to perform this work. And last, but of course not least, I would like to thank the organizers for being given the opportunity to share some of my work with you today. Again, here is some of my information, including my email, if anyone in the audience would like to ask any questions. And of course, all that is left to do is to thank you for listening.